Hi guys, welcome back to another episode of Chamber's Guide to Not Effing Up A-Level Biology. Today we're going to be doing the Calvin cycle. Now this is going to be the second part of our photosynthesis subtopic of like the respiration topic. I suggest you watch the video about the light-dependent reaction before we begin talking about the Calvin cycle. The main thing you need to know about the Calvin cycle is that it's simply a series of biochemical steps in order to produce sugars. And I know the numbers and stuff can get really confusing, so we're going to slowly build it up as we're going through the cycle itself. So let's get started. The first thing I'm going to talk about is a few basic principle key ideas that you need to know before we begin talking about the Calvin cycle. So here, I'm going to draw a thylakoid. So I'm going to put phi over there, thylakoid. And here is your chloroplast. Poor drawing, but nevertheless, here we are. So you have the thylakoid over here, and the fluid surrounding the thylakoid is the stroma. Now, I know I haven't drawn out the granum and all the other weird stuff, but this is effectively to illustrate a few simple points. The first thing that you need to know, and this is absolutely key, is that the Calvin cycle happens in the stroma. The Calvin cycle happens in the stroma. I don't know if you have to do a daily mantra to try and remember that, but you have to remember that it's in the stroma. It's not in the thylakoid, it's not in the thylakoid membrane. I know when I did this topic, I had a bunch of weird assumptions, and you honestly just need to drill it into your head. The Calvin cycle happened in the stroma. The second point I'm going to talk about is the fact that the stroma is close to the thylakoid. Now, when we were doing the light-dependent reactions, we were talking about why we're producing NADPH. Why are we producing so much of it? And we'll find out that in the Calvin cycle, NADPH donates electrons, and it's such a good way of helping synthesize sugars. So because we're producing a lot of NADPH here, it can easily diffuse into the stroma because the thylakoid is effectively within the stroma, so it makes it a lot easier. So I think we can write as thylakoid, say surrounded by stroma. Therefore, NADPH can easily diffuse into stroma. That's our second point. The last point I'm going to make is that in the Calvin cycle, we have a bunch of different enzymes and substrates and different things that are going to start colliding with each other in order to react. So the third point we want to make is that the stroma contains the enzymes required for the Calvin cycle. So I'm going to go over those 3 points again. First one, key point, you really need to remember this. The Calvin cycle happens in the stroma. Second point is that the thylakoid is surrounded by the stroma, therefore NADPH can easily diffuse into the stroma. And the third point we have is that the stroma contains the enzymes required for the Calvin cycle, so you can form a lot of those enzyme substrate complexes in order to help the Calvin cycle move on. Now, the next part I'm going to do is I'm going to talk about the Calvin cycle itself, and I strongly suggest you look at the textbook first and try and familiarize yourself with the different things that are happening before you look at this video, because this is where it gets quite weird and it can really get confusing. I remember when I did the Calvin cycle and my brain was exploding because it was really hard, and that's totally okay. So the first thing I'm going to do is write out IUBP. I'm going to draw an arrow from IUBP to an unstable 6-carbon molecule. So I'm just going to write that out. Unstable 6-carbon molecule. Now, I'm going to denote the number of carbons with a red and an orange pen. So I'm going to draw out the number of carbons for IUPP, which is 5. And in orange, I'm going to write it out as 5C, symbolize the 5 carbons. Alright, let's get started. So in a plant, in order for the Calvin cycle to take place, we need carbon dioxide. And that carbon dioxide can diffuse into the stroma. So, you know, just as changes in pressure, it could just be like a potential gradient for the carbon dioxide to enter the plant, or it could just be from a byproduct from respiration. Respiration does not equal photosynthesis. I know it sounds obvious, but when doing this topic, you can get those really weird assumptions and start dragging different things together, because the molecules can be like they have the same name, and you know, it gets really weird. But do remember, photosynthesis is not respiration. Respiration is using up energy, and photosynthesis is creating a sugar, a storage molecule, in order to store that energy. All right. So as we talked about the diffusion of CO2 into the plant, I'm going to draw an arrow from outside, and I'm going to call it CO2. And that carbon will fixate onto the IUPP. So I'm going to write out what you need to know. So 1, IUPP fixates onto a molecule of carbon dioxide. Now, this is the first part of our little acronym, and I'm going to just put it over here. I don't know if you've heard of it before, but it's called PHI-RE-RE. So PHI-RE-RE. And currently, we're in the phi part, which is fixation. So this is the key thing you need to know. IUPP fixes on to a molecule of carbon dioxide. By fixing, we mean that it's essentially bonding onto the IUPP. Now, the IUPP has 5 carbons. We're adding a carbon from the carbon dioxide, so you end up with an unstable 6-carbon molecule. You don't need to know the name of that molecule, but I'm just going to draw it out here. So, in red, 1, 2, 3, 4, 5, 6. 6 carbons. Now, unstable because it's not stable, and it's going to break down really easily. In the world of chemistry, things can break down depending on how stable they are, how strong the bonds are, and essentially the fixation of carbon makes a 6-carbon carbon molecule that's really unstable, and it will split easily. So the next part that I'm going to do is that I'm going to break up that 6-carbon molecule, so I can put that in black. And it's going to break down in the middle. I'm going to form 2 molecules that are 6 carbons each. 2, 3, 4, 5, 6. And this molecule is called G3P. The whole name, I think they can sometimes put it up in the exam, so I'm going to call it glycerate 3-phosphate. I know in some textbooks they're like, oh, it's called PGA, it's called... This is the thing I don't like about this topic. There's so many different conventions of writing stuff, but this is what you need to know for our syllabus. So you form 2 molecules of G3P. And I'm going to put, like, over here, 2 molecules. Just so that we can really make sure that we're understanding the idea that one molecule is breaking down into 2 molecules. I think, just to make it a bit more clearer, I'm going to do that in black, just so we can still stay on that colour scheme. So, 2 molecules. Over here, we have 1 molecule over here, 1 molecule over here, and 1 molecule over here. So let's go through that one more time. The 1 molecule of IUPP fixes on to 1 molecule of carbon dioxide, forming 1 molecule of an unstable 6-carbon molecule. And as it's so unstable, it will break down into 2 molecules of G3P, or glycerate 3-phosphate. So I'm going to write that down over there. So we've talked about the fixation of IUPP. So forms unstable 6-carbon molecule, easily breaks down, forming 2 molecules of G3P. Just going to highlight the important parts. So, unstable 6-carbon molecule easily breaks down, forming 2 molecules of G3P. This is where it gets interesting, because this is where we start talking about those molecules of NADP. Remember, we're talking about photosynthesis, so we always need that P. So the next step is going to be simply a series of oxidation and reduction steps in order to make something called triose phosphate. Now, triose phosphate is another 3-carbon molecule, and triose phosphate can be used to form sugars like glucose, sucrose, things like that. So draw another arrow, and we're going to go over how NAD works here. So NAD, remember, it's an electron carrier, and NADP, even I need to get the hang of it, NADP is in its oxidized state. So NADP can accept electrons, NADPH can donate electrons. So in the light dependent reactions, we are forming NADPH. So I'm going to put that over there. NADPH. And NADPH, as I said earlier, it will donate electrons. So that will go from NADP plus and an electron. In addition to this, we also need ATP. So I'm going to draw another arrow here. So ATP will go to ADP and our inorganic phosphate. The ATP essentially provides the energy for this reaction to happen. It doesn't phosphorylate the G3P, it doesn't do anything weird like that. It essentially is just there to provide the energy for the reduction of glycerate 3-phosphate. In other words, the glycerate 3-phosphate is going to gain an electron. Now, for every molecule of G3P, you need 1 molecule of NADPH and 1 molecule of ATP. So in this case, because we formed 2 molecules of G3P, we need 2 of those NADPHs and we need 2 of those ATPs in order to balance everything out. So I'm going to put a 2 there, I'm going to put a 2 there, and that forms our final molecule that we're looking for, which is triose phosphate. So TP, triose phosphate. Triose phosphate has 3 carbons as well. 1, 2, 3. And remember, because we have 2 molecules of G3P, we're going to be forming 2 molecules of triose phosphate. So I'm going to put 2 molecules there. And just to reinforce the idea of the number of carbons, 3C goes there, and a 3C goes over here. The next part is, in order to complete that cycle, we need to reform IUPP. Now this is where it gets confusing, because in order to reform IUPP, we always need to have 5 carbons back. Now we have 3 carbons over here, so essentially we need to get 2 more carbons from somewhere. And what happens is that we need to recycle a bunch of triose phosphate in order to form IUPP. So let's rephrase, let's go back a bit, and let's start talking about this. So when we're looking at the calculations involving the Calvin cycle, IUPP will always be 3. Now you must be wondering why I wrote 1 over here, 2 over here, and that's to illustrate the important point that each G3P needs its own NADPH and ATP, how much that unstable 6-carbon molecule will break up, and that will essentially form 2 G3P. So you must be thinking, oh, this is really weird, why are we going back? But this is essentially reinforcing, pardon me, the important point over here about the proportions of how they're breaking down. And now we can start talking about the actual numbers. So whenever we're trying to do a balanced Calvin cycle, the number of IUBP should always be 3. I'm going to draw a circle around that. Now, if we have 3 IUBP and we're trying to form a 6-carbon molecule, we're going to need 3 carbon dioxides, right? Because we have 3 IUBPs here. Each carbon dioxide will bind to 1 IUBP and will essentially form 3 6-carbon unstable molecules. So I'm going to turn that into a 3. It's going to fixate, and we're going to form 3 unstable 6-carbon molecules. Now, because it's going to break down, each 6-carbon molecule is going to break down into 2 glycerate phosphates. So if you do the math, we're going to have 6 glycerate phosphates. So I'm just going to change that over here. Make that into 6. Now, we know that each glycerate 3-phosphate requires its own NADPH and its own ATP. So I'm going to change these numbers as well. So that's going to be 6 because we have 6 molecules. So each of them will require their own NADPH and ATP. And finally, we'll end up with 6 molecules of triose phosphate. So in these 6 molecules of triose phosphate, let's count out how many carbons we have. So each triose phosphate has 3 carbons. And we have 6 molecules of triose phosphate. So 6 times 3 gives us 18 carbons. So I'm just going to draw it out just to make it a bit easier to look at. 1, 2, 3, 4, 5, and 6. Within these 6 molecules of triose phosphate, 1 of the triose phosphates will exit the Calvin cycle, and 5 of the triose phosphates will react to reform 3 molecules of IUBP. Why? It just does. And if we count out the number of molecules, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 carbons, how many carbons does 3 molecules of IUBP have? 5 times 3 is 15. Those 5 triose phosphates will reform into 3 IUBPs. Now, in order to form bonds, we're going to need energy. And that energy is, again, going to be provided by ATP. But this time, because we've balanced our cycle out, it's going to require 3 ATP. Why? Because we're forming 3 IUBPs. So I'm going to go over that one more time because I know that's really confusing. So over here, when we're looking at how much ATP and NADPH we need to use, for each G3P, we have to have their own ATP and NADPH. That's just a thing for G3P. When we're reforming IUBP, however, the number of ATP molecules refers to how much IUBP we're forming. So in this case, we're forming 3 IUBP, so we're going to require 3 molecules of ATP. So I'm going to start writing everything down over here. So 1, IUBP fixes onto a molecule of carbon dioxide. 2, forms an unstable 6-carbon molecule, easily breaks down to form 2 molecules of G3P. Now, what I'm going to do is I'm going to start changing all of the numbers here because I want to move over that balanced Calvin cycle onto what I'm writing over here. So over here, 3 IUBP molecules fixes onto 3 molecules of carbon dioxide, forms 3 molecules of an unstable 6-carbon molecule, easily breaks down to form 6 molecules of G3P. The next part we're going to write is 6 NADPH and 6 ATP are used to reduce the G3P into 6 molecules of triphosphate. And this forms as a byproduct ADP, 6 ADPs, 6 inorganic phosphates, and 6 NADP pluses. The NADP pluses will now return to the light independent reactions in order to collect more electrons and essentially be recycled over and over again. Number 4, the triphosphate. So one triphosphate will exit Calvin cycle. 5 triphosphates will react to reform 3 RUBP. This requires 3 ATP, as number of ATP refers to number of RUBP formed. And this goes back into our acronym. The first part is called fixation. The next part over here, as we talked about reduction, reduce. So that's the second part. So this is your fixation part. This is your reduction part. And finally, we have the final part of our acronym, which is the regeneration part of RUBP. Now, one key point that I've missed out from this diagram, and you'll know from the textbooks, is that I haven't mentioned the enzyme. And I specifically left that out for last because I lost a lot of marks for not talking about the enzyme. So the enzyme that we need to know is Rubisco. And Rubisco catalyzes the reaction between RUBP and carbon dioxide to form that unstable 6-carbon molecule. So that means we have to edit our explanation over there. So I'm going to go back over here. We're talking about the fixation of RUBP. So 3 RUBP fixates onto 3 molecules of carbon dioxide. This reaction catalyzed by Rubisco. It's a really important thing that we need to remember when talking about the Calvin cycle itself. Now, this one triphosphate that we formed, this whole cycle, we're going to have to do it one more time, because by doing that, we'll be able to form 2 free triphosphates. And glucose is a 6-carbon molecule, so those triphosphates can react to form glucose. And as plants, we know that those glucose molecules that can form large polymers that are insoluble, like amylose and amylopectin, and that effectively allows glucose molecules, or the polymer of glucose, to behave as a storage molecule. So really complex video, a lot of weird stuff to remember, but as long as you remember these numbers over here and the fact that this cycle needs to run the whole thing one more time, you should be fine. Apologies for my throat today. I hope that was helpful, and I'll see you guys in the next video. Bye.